Hi, everybody. Thanks for joining us today. And good morning, good afternoon, and good evening. I can see from the attendee list, we've got quite a range of locations for where people are from. So, whatever time of day it is, I really appreciate you joining us for the webinar today. So, the title of today's webinar is Count Cells Without Slides, Automating and Optimizing Cell Counting. My name is Andrew Jones, and I am the Market Development Manager with DeNovix. I'll be moderating today, and presenting will be Corey Pezak, who is one of our application scientists with DeNovix. And he'll be taking us through how to standardize counts of tissue culture, primary cells, nucleic acids, and other complex samples on the CellDrop Automated Cell Counter. The webinar will look something like this. We will have an introduction to the technology of CellDrop, which will take about 10 minutes, just for those of you who aren't familiar with it already. We'll then switch to a live demo. So Corey is based in our Wilmington, Delaware headquarters, and he's in the lab at the moment. He'll have some live cells that he's going to run to show you some examples of how to do that on the CellDrop. That'll take about 10 minutes. And then we'll also finish with a Q&A session. So for the questions and answers, you will see in the Zoom interface, you have a Q&A option in there. During the course of the webinar, if you have any questions, please feel free to add them to that section. We'll monitor them, we'll answer them as we go along, and any of them that are relevant to the wider group, we'll put those to Corey when we do the Q&A. So before we get started, I'll just do a quick introduction to DeNovix in case you're not familiar with our company. We were founded in 2012 by Fred Keelhorn, and Fred was one of the original founders of Nanodrop Technologies, if you're familiar with that technology. We're based in Wilmington in Delaware in the US. So all of our product development, all of our application support, all of our manufacturing service is all based at our HQ in Wilmington. We're the inventors of multiple award-winning instruments, and we're proud to have some of the most highly reviewed instrumentation on the Select Science web portal, which is where we choose to do to collect our reviews. We have over 12,000 citations and 1,000 plus reviews from researchers around the world, which we're very proud of. We like to think this is a combination of great instrumentation and our focus on providing the best technical support we can. So we're laser focused on being one of the best in the industry for supporting you. Our product portfolio has three main pillars. So our original product line was the DS series, and that's a very successful micro volume spectrophotometers, fluorometers, and combined instruments. The CellDrop Automated Cell Counters are going to be the focus of today's show. So I won't talk too much about those because Corey will give you the introduction. And we're also very excited and very proud to announce our latest product that we have just launched, which is called the Squid Pipette. And the Squid Pipette is a unique instrument. It's the first electronic pipette that allows you to aspirate the full volume of 1 to 1,000 microliters in a single device. So if you're interested in that, lots of information on our website, but that's not the focus of today. But feel free to reach out for more information on that if you're in the market for a new pipette. Today's speaker is Corey Pezak. Corey is a member of our application support team based in Wilmington in Delaware. As you can see, Corey is in our lab at the moment. Corey has a master's degree from Villanova in biological sciences and a specialty in molecular genetics and immunology. And Corey's focus day to day is largely just providing technical and application support to our install base and to anyone currently running free trials on our instrumentation. So with that, I'm going to hand over to Corey.
Thank you, Andy, for that introduction. Before we begin, I just want to tell a very quick and brief story. So during my graduate studies at Villanova University, my research focused around macrophages, and I begin by counting them manually with a hemocytometer. And I really wanted something to streamline that process because, let's be honest, it is a very laborious process. Luckily, my neighboring lab has a CellDrop, and I was able to use it. And I don't want to say this instrument's the reason why I finished my project 3 months early, but it had a big part in it. So, enough about me. Let's get into why you're here. So you may be asking yourself, how does this CellDrop count without slides? First and foremost, it's the direct pipette technology because it eliminates the need for expensive plastic slides. This instrument has dual fluorescence and bright field technology, which is great for trypan blue and acridine orange and rubidium iodide counting. It has a suite of apps for multiple workflows and an HD touchscreen, just like your smartphone or tablet. And finally, it has flexible connectivity to export your data in multiple ways. Now, to dive deeper into the direct pipette technology, the upper and lower sample surfaces are optical grade sapphire surfaces, so they're very robust and scratch resistant. And you could think of the CellDrop as a hemocytometer microscope hybrid, and you'll see more during the live demo. So as the animation shows, once you pipette your sample between the two surfaces, surface tension holds the sample between the chamber. Now, we do have 3 chamber heights that are controlled by a precision screw, which I'll talk about next. So altogether, this technology is easy to keep clean with no sample carryover and no calibration required. So there are benefits of the direct pipette technology. First and foremost, it's environmentally friendly because, again, you do not need to dispose of any plastics. Now, a few benefits of the chamber heights. So one, there's no need to dilute or concentrate your samples because we have a wide density range of 700 cells per milliliter all the way up to 25 million cells per milliliter. And we also have a large cell size range from 4 microns to 400 microns. Now, I would like to focus on the chart at the bottom of the screen. As you can see in that middle column, 100 microns, that's our standard chamber height. But to the left and to the right of it, we have 2 other chamber heights for higher density samples and lower density samples. Now, the CellDrop has 2 modes. We have bright field, which is great for clean tissue culture samples, which you can use trypan blue or erythrocin B for viability assays. We also have dual fluorescence for primary cell samples, or if your cells have a funky morphology, this is great to use fluorescent assays like acridine orange rubidium iodide, or if you have a GFP tag inserted in them. Now, I want to talk a little bit about the assays that I'm going to demonstrate on the CellDrop. First is the bright field viability assays of trypan blue and erythrocin B. Now, they both they stain dead cells in a dark color, and they permeate into dead cells and bind to negatively charged molecules. And they're very commonly used in manual cell counting. And if you haven't heard of erythrocin B, don't worry. It is new, it is not toxic, and it's better than trypan blue. Now, let's switch gears to fluorescence counting with acridine orange rubidium iodide, which is commonly referred to as AOPI, and I will call it from here on out. So they are nucleic acid binding dyes, very, very specific. So AO permeates the cell membrane of both live and dead cells, and it fluoresces green. So you can think of green live. And then PI cannot permeate live membranes and only enters compromised dead or dying cells and fluoresces red. So red dead. And this is because there is a fret quenching in dead cells that causes PI to absorb the AO fluorescence signal, ensuring precise counting. So you may be asking yourself, when and why would I use fluorescence counting? So again, for primary samples like the top image that are littered with red blood cells, this stain is great because trypan blue is a blanket assay and will non-specifically bind to debris and non-nucleic cells, such as red blood cells, whereas AOPI will specifically bind to live and dead nucleic cells and not staining red blood cells. So I want to talk about a little bit of the data between the two assays and when to use them. So the top graph are CHO tissue culture cells. And as you can see, there is a one-to-one comparison with trypan blue and AOPI. But the bottom graph are primary PBMC samples. And as you can see, trypan blue overcounted by a factor of 2 when you're comparing them to AOPI. And this just means AOPI is a lot more specific.
Hey, Corey. Okay, great. Thanks. So we've got a couple of questions that have come in already. If you do have more questions, as I mentioned, put them into the Q&A box and we'll pick them up as we go along. Just a couple of questions. First one that we had in was whether the cell drop has to be connected to the network in order to analyze cells.
Yeah, great question. It does not for a few reasons. The first one is all the data is stored on the instrument itself, and then you can always export via USB. So there's no cloud-based storage.
And all the analysis takes place on the instrument as well, of course. The other question was, why does erythrocin B work better than trypan blue?
Yeah, so erythrocin B is superior to trypan blue, like I said, because it is non-toxic. It has a longer lasting stability when stored and enhanced optical contrast for automated counting systems, just like the CellDrop.
Great, thanks, Corey. That's all the questions we've got for now.
Wonderful.
Thanks.
So now I'm going to be switching to a live demo. I'm going to be using the CellDrop FLI, or the fly, as we like to call it. We'll be doing 3 counts today. First one is going to be jerk hats with trypan blue. Then I'm going to switch to fluorescence counting. The second count is going to be jerk hats with AOPI, and then primary PBMC samples with AOPI. So here's an overview of the instrument. As you can see, this screen here is a 7-inch HD touchscreen, again, just like your smartphone or your tablet. And we have the 3 apps right here. We have trypan blue, AOPI, and primary cell AOPI apps that we're going to be using today. So as we move up, you're going to see where you load your sample. And as you can see here, once you lift the arm up, we have both sample surfaces. So we have the lower one here, and we have the upper one up here. So to load your sample, you're just going to put the arm down. So I'm going to open up the suite of trypan blue apps here, and I'm going to select the tissue culture app. So now I'm just going to do a little bit of a mixing of our cells, mix them with trypan blue, and then I will be loading onto the instrument. And now I'm doing a 1-to-1 mixture of 10 microliters of cells, 10 microliters of trypan blue, and then I'll be loading onto the instrument. And then to load, what you'll do is there's 2 notches on either side. You use them as a guide for your pipette tip up to where the sample surfaces meet and pipette normally. So that's my favorite part. This is a live feed. Watching all of your cells go across the screen is my favorite part. So what we're going to do now is we're going to check the exposure. As you can see, we have different exposure settings, and there's going to be these information boxes on the bottom left of all of these windows just to show you if you're correctly exposed or not. And that's the same thing with the focus. Now everything is set correctly. I'm just going to press count. And within a few seconds, we're going to see not only a picture of the cells on screen, but we're also going to see some data on screen as well. So as you can see on the left-hand side, we have live cells per mil, dead cells per mil, total cells, and a viability percentage. And then if you move down here, you can see some nominal cells as well. So cleaning the instrument is very quick, very easy. You're going to take a Kim wipe, fold it up a few times, and once you lift the arm, data is saved automatically. And again, these surfaces are optical grade sapphire. They're very robust. So you could put a little bit of pressure on them. You will not break them. And just like that, we clean the instrument and we're ready for our next count. So now I'm going to be going to our AOPI counting with our AOPI app, since we're going to be using jerk hats. And just like with trypan blue, I'm going to be mixing some cells with our AOPI. Now, even though it's a fluorescent stain, a lot of people do have that question of, do I have to incubate my cells before I load them on the CellDrop? And that is no. We do recommend to mix them and load them right away. And just like that, you're going to see your cells come across the screen again. Now, with AOPI, we have 3 different channels just because it is fluorescent. We're first going to go on the green channel. We're going to check the exposure. Again, there's an information button here and the focus in the green channel. Then we're going to move over to the red channel and make sure that the exposure is set properly as well, and only the exposure. Everything is set, and now I'm going to press count. And you may have noticed I did not change the focus and exposure settings, and that's because the instrument remembers all the settings that you previously inputted. So that means a very quick workflow that you don't have to change settings between counts. And just like the Trypan Blue app, you're going to have your live cells, your dead cells, and a viability percentage with nominal data. Now, if you need to gate on the fly, you can do so. If you touch this button down here on the bottom left, a graph is going to pull up, and you can use this sliding bar to gate your minimum or maximum size. And then if you do so, you can apply those gatings to result, and the live and dead cells will change. Again, cleaning just like the Trypan Blue app, we're just going to firmly press and wipe the top and bottom sample surfaces. So now I'm really going to show you why AOPI is so valuable, because I'm going to do a primary cell count with PBMCs in whole blood. So again, just doing my 1-to-1 dilution. We're going to mix really quickly, and we're going to load. And as you can see on screen, there's a lot of darker cells. Those are the red blood cells. And as you can see in the green channel, we do have a few green dots. Those are our live cells. And in the red channel, you will see some dead cells. And then we're just going to go ahead and press count. And again, within a few seconds, we're going to get an accurate count of live and dead cell samples within the sample. So as you can see here, all of the darker cells are red blood cells, and they're not being picked up, but these green ones are. And if you just want to check and make sure which cells are live and dead, we have this channel button right here, which you can look at your cells in any channel that there is. So I want to look at the green channel. I could do so here and make sure my green signature is being picked up. And now if you want to optimize any settings, you could do so on the fly as well with this optimize settings button. If you change any of these parameters, you can count with the current settings, and then a green box over here will pop up saying save settings to protocol. So you don't have to write anything down or remember them. And there's 2 ways to export data. The first is in this app, and the second is in the data app that we have. So if you hit this export button, you can see the items you can export. So maybe I just want the CSV. And then once I press next, there's the different destinations. So we do have USB, or you can export via email, network folder, or network printer. Now, I do want to show you the data app. So as you lift the arm and you wipe away, your data is automatically stored. So once you open the data app, you'll select by app, which I'm going to go into AOPI. You can select by a certain protocol if you work with one protocol, or you could recall a week's worth of date if need be. So I'm just going to take this count, add it to a report because I don't want all of these counts. And then I'm going to click here. And if I were to press that export button, it would be the same way as if I were in the app. Another cool feature that we have, if you press on results list and go to graphs, we have a lot of different graphs that we show here on screen. So now that I'm done with the demo, I do want to talk a little bit more about the CellDrop itself. So we do have 4 models. We have a fluorescence and bright field model and a bright field only model. we have a 4x objective that can count cells between 4 and 400 microns, and a 10x objective that can count between 2 and 20 microns. And we can support a wide variety of applications such as protoplasts, stem cells, algae, and even yeast. And along with our regular counting apps, we've also developed 2 machine learning apps. So we have an organoids app and a hepatocytes app. So the organoids app was designed for organoid, spheroid, or tumosphere cell types. And this app reports counts, size distributions, and density. And then the hepatocytes app is great for drug metabolism and toxicology workflows. So this app reports live and dead hepatocytes along with leukocytes and debris. Now, the counting algorithms have already been trained by me and the rest of my colleagues, so it does not need to be trained on your samples. If you're sitting there right now and you may work with samples that are biohazardous and you need to use slides in your workflow, you can still use slides on the CellDrop. We do have a slide mode. You can use our DeNovix reusable slide or select disposable slides. So now that I've talked about the instrument, I want to take this time to thank everyone who has not only bought our instruments, but even taken the time to review our instruments. So without each and every single one of you, we would not be able to continue to create instruments that sit at the forefront of cutting edge research. To wrap up this webinar, I want you all to leave with 2 things in mind. First, that the direct pipette technology is unique because it reduces plastic waste, it has cost benefits, and it's fast and easy to use. The second thing I want you to walk away with is the performance of the CellDrop. So the 3 different chamber heights allow for a wider dynamic range. It is feature rich and produces high quality and accurate results. And finally, it is fully networked.
Thanks, Corey. We've got a lot of questions come in, so maybe we can make a start on those straight away. Okay, so first question was that you mentioned algae. Can you do native fluorescence of chlorophyll?
Yeah, that's one of the cooler applications that our customer use this instrument for. So we do have customers that use the CellDrop to count their intrinsically fluorescent chlorophyll samples. And we do have tech notes on our website that describe how we can do that.
Cool, thank you. Question about two kind of software ones. How many samples can you store on the instrument? And does the instrument allow for multiple user accounts?
That's a great question. We do not have a definitive number of counts that can be stored on this instrument, just because it takes into account not only counts, but also images, but it does have 256 gigabytes of storage. And we have a user's accounts app that can input a lot of users within the CellDrop.
I mean, it's in the thousands of samples, right?
Mm-hmm.
Okay. Can you graph multiple samples together? So you showed the graph questions. Can you overlay them and have multiple samples in those graphs?
Yeah, you can have, for the graphs feature, you can see different samples side by side with all of the different features that we have. So we have live cells, dead cells only, viability, size.
But a couple of questions about the PBMC samples. First question was which dye is the best for counting PBMCs? And I think it's probably the one that you were using, which was AOPI, right?
Mm-hmm. Yeah, not trying to be biased, but yes.
Yeah, no, I think it's reasonable. I think it's a good dye for that. The other question about PBMCs, Kieran, asking about all of the cells in that particular image that weren't counted. There were a lot of cells in there that weren't counted. And the question was, what were they and why weren't they counted?
Yeah, so all the darker cells in the background that were not counted that you all saw were red blood cells. And that's just because they're non-nucleic cells, and AOPI does not bind to those cells.
So we've got a little more come in as well. Let me just catch up on these. There was a question about the machine counting of organoids. Is it a specific type of organoids? Does it depend on size, or is it trained on multiple species, multiple types?
Yeah, that's a great question. So the organoids app does depend on size. We have two different chamber heights for that app. We have a 200 micron chamber height and then a 400 micron chamber height. I would definitely say the max organoid that we can count is getting up to the 400 microns in diameter. But if you are definitely interested in learning more about that app, we do have a tech note, or you can reach out to me and one of my colleagues.
Cool. There was a question from a current user about how you change the chamber height. And if you could explain that. I don't know if we can switch back to the instrument or not, whether we have that set up.
Yeah, so we could definitely show this screen again. So it would be in the protocols section of the app. So I'm just going to pick the Brightfield app. And then once I get onto protocols, if everybody can see my screen, I'm going to hit this plus button here to create a new protocol. And then towards the bottom left, you will see chamber height. So once you select one of these, maybe the 400 or the 50, and then you're going to have to save that protocol and then switch to that protocol because it will always go to the default. Once you do either the 400 or the 50, you'll see the arm either raise or lower. And that's how you know that you change your chamber height.
Perfect. So, Corey, before you get out of that, yeah, too late. There was a related question that I've just seen that was about how you handle non-circular cells. This is probably a good screen to to demonstrate that.
Yeah. So we have an irregular cell mode. So a lot of automated cell counters are produced to count circular or near perfect circular cells. The CellDrop can get away with it a little bit, and we have an irregular cell mode. This information button is really good. It shows you two pictures of what an irregular cell mode looks like. And even with this little bit funky morphology, we're still highly accurate.
Mhm. That's great. I had a couple more questions on cleaning and what is the required periodic maintenance? So between samples and periodically.
Yeah, this instrument's really maintenance free. As you saw between samples, I was wiping away with a Kim wipe and my sample surface was clean. If there is anything on your sample surface that's left behind by any debris or anything like that, you can pipette in any alcohol of your choice or bleach, just like you would if you were loading a sample, and just wipe away, and that sample surface will be clean.
There's a couple that I'll group together here, which are asking about how the cell drop deals with clumping cells or aggregated cells. So there's one question about having particularly sticky cells. How well does it work with them? And also, can you see how big the clumps are? Do you have do you have any way of seeing what the cluster size of cells is?
Yeah, so the CellDrop has a declustering algorithm that it's bread and butter. So once you count cells, this algorithm can pick out single cells in a cluster of cells and count those as one. A few apps do report cluster measurements as well.
So it'll give you a breakdown of clusters of one, two, three, four, five cells.
Correct. And you can see that in the CSV file or in the PDF file.
Great. There's a question about using trypan blue or erythrocin B and whether the dilution is taken account of, so diluting it with the reagent itself, or do you have to do that afterwards?
Yeah, that's a great question. So we already factor in that one-to-one dilution into the protocol. And that's a nice thing about you can create your own protocol. So if you do any external dilutions, you can add that into the protocol and the instrument will do the math for you. So there is no need for external math.
So there's a question about optimizing protocols and whether there is a sort of a stepwise process to go through to optimize a protocol. If you have
Yeah,
any
so each
comments on
app, yeah, so each app has its own protocol settings that you can optimize. but I'm going to say the easiest thing to do is to reach out to me or one of my team members. We can easily optimize a protocol for your cell type. And then we send that protocol back to you. So you can input it on your instrument and you don't have to worry about playing with the settings and making sure it's working properly.
And it's fair to say, I think, in my experience, that the default protocols get you a long way, right? So the default protocols are really well optimized for the majority of cell types that we use. And then for anything else, we kind of have the options in the software that you can use to finesse optimization. Do you have a way to update the apps on the instrument over time?
Yes. So all of our software updates are free. They come out every 6 months or so, and you can update the instrument in two different ways. First is downloading the zip file from our website onto a USB, or if you're able to connect the instrument to their network via Wi-Fi or Ethernet, you can download the update through there.
Great. We also have a question about whether the media affects the AOPI stain.
Yeah, that's a really good question. The only media as of right now that can probably have a little bit of a problem is RPMI media, and that's just because of phenol red. But there are some workarounds for that. You can dilute your samples into PBS, and your AOPI signature will still be strong.
Perfect. And I think the final question that I can probably take here, it is about calibration and whether it needs an annual calibration.
That's a great question. So this instrument is calibrated for life just because there is an initialization process that happens each time that you turn the instrument on. That process just makes sure the software and hardware are working together and communicating properly.
Perfect. Thank you, Corey. There are other questions. So some of them are very specific to different cell types. So well, what I might do is say that we'll wrap up the Q&A there. If we haven't got to your question, I promise we will absolutely follow up with you. Either Corey or one of the apps team will definitely answer your question for you. After the webinar, we will send you an email with a link to a recording as well. So if you want to share it with your friends or look back at any of the particular details, it'll be there. We'll also put a little survey in there. We'd really appreciate it if you fill in the survey because it helps us to develop content that works better for you. If you have any questions, please feel free to get in touch with us. All of our details are on DeNovix.com, of course. And if you're interested in the new squid pipette, all of the details are on the website as well. So I would just like to say thank you again, Corey, and thank you to everyone attending today and for a lot of great questions. Really appreciate it.